Good afternoon. My name is Ron Hawkins. I'm with the San Diego Supercomputer Center, and I am the director of our industrial engagement programs, but I am also managing our campus HPC cluster. And in that capacity, I'm going to talk to you today about a project we have called BioBurst, which is where we're trying to leverage the DDN IME technology to accelerate bioinformatics computing. And so first I want to quickly acknowledge the National Science Foundation, who provided the Campus Cyber Infrastructure grant that funded the equipment purchases that I'm going to talk about in connection with this project. And so SDSC operates the Comet and Gordon supercomputers for the National Science Foundation, but we also have sort of a medium-scale campus HPC cluster called Triton Shared Computing Cluster, TSCC, that we operate for researchers on the UCSD campus. And this is mainly what we call a condo computing, where the researchers purchase compute nodes, contribute them to the cluster, and then they can run on their own nodes or kind of get the run of the whole cluster. And it's sort of 300 nodes, 6,000-ish cores right now, not including the GPU cores. And so many of the UCSD researchers can get access to, and they do get access to, NSF computing resources or DOE resources. But we saw a need for another class of system where there was a quicker, quicker access without proposals, without wait times. And so this is the genesis of the campus cluster that we started about 4 years ago. And the condo part of it gives most researchers access to a bigger cluster than they could get just purchasing nodes for their own lab. And so this is just sort of how that condo model works. So they buy the nodes with equipment funds from grants or startup funds, and then they pay a one-time fee that covers the common infrastructure like the home file system and the switches. And then, like it shows here, then they can, if they buy one node, they can run on the whole cluster. They burn their annual allocation of time up faster, but it's their choice. And then we have a section, we call it, it's kind of corny, we call it the hotel section, but they can buy time by the core hour, 2.5 cents per core hour. And so TSCC, it's pretty heterogeneous because we've been adding to it for 4 years. So it's got 3 generations of Intel processors, 16 to 28 cores per node. It's got a mixed 10 gigi and QDR InfiniBand interconnect, and it's got a mix of NVIDIA GPUs, mostly gaming class GPUs, but I think we have some K80s in there. And so the idea behind this Campus Cyber Infrastructure, CCSTAR proposal, was that we have a very vibrant life sciences computing enterprise at UC San Diego. We have a rapidly growing bioinformatics workload, and we wanted to bring in some technology that we thought could accelerate those workloads and both improve the productivity of the life sciences researchers, but then also free up more of the cluster for our other researchers. And so this is where the IME technology comes in. We made this proposal, and we got the $500,000 grant to buy the equipment I'll talk about, and that started earlier this year. And a couple of the selling points in our proposal were, one, we wanted to show that we could leverage technology that was being created in the exascale program, i.e. burst buffer technology, into campus research computing kind of environment. Now, we're not using the IME as a burst buffer, but the basic technology is the same. And also, we wanted to show that this could be a test bed for scaling up to bigger NSF systems and our own campus system if this sort of modest capability proved out. And so through our own experience and also other published research, and the references are at the back of my proposal, we know that many of these bioinformatics codes do a lot of small block, small file, random access types of I/O. And so we wanted to bring in some technology that could hopefully address some of those issues. And that was really the big core part of the proposal. And so with the grant funds, we bought kind of 3 big, not big, but 3 separate components. And so we bought a 2-node IME 240 system with 40 terabytes of usable NVM. We bought an FPGA-based computational accelerator system from a San Diego company called Attica Genome. It's called a Dragon system, and it's specifically for genomics and transcriptomics acceleration. And so it can do things like, take like a whole genome sequence alignment and variant calling that would take 10 hours on a normal compute node and do it, in like 25 minutes. And then we bought some 24 additional computing nodes for the cluster that gave us, sort of a separately scheduled resource to run the codes on in kind of a sandbox for this whole part of the cluster. And so this just shows you, kind of what I just talked about, how things are arranged. So we've kind of got the TSCC cluster, and it's wired into our campus 10 gigabit fabric, as well as connections to high bandwidth offsite networks like Scenic and Internet 2 at up to 100 gigabits. We've got a 650 terabyte Lustre file system, 40 terabyte home file system, ZFS based. And then so the new BioBurst piece kind of logically sits off to the side there, connected up with the IME system and our Lustre system. And so this is just a little more detail there. So we've got the 24 new nodes are all connected to an EDR 36-port managed switch. And then with the IME 240s, we've got a total of 4 EDR ports that go into that same switch on the client for the client-side communications. And then on the other side, we've got 2 40-gig Ethernet ports that tie into our data center switching fabric, which is 2 512-port Arista 7508 switches. And then those tie into the Lustre file system. And so, we operated the Gordon system for we're still operating it. It's been over 5 years. It's got 300 terabytes of flash memory in it, and we've gotten a lot of experience, deploying that flash memory for different scientific computing work cases and gotten some good results. But the thing about that was that there was a lot of human-in-the-loop interaction with the Gordon, what we call the I/O nodes, to configure those for each science user that came along. So the allure we saw with the IME was to put some automation into that, where we could the client side would sort of talk to the IME under the hood and buffer the working sets into the NVM, where we could get high IOPS read-write, and then the IME would take care of syncing any writes back out to the Lustre system, so we had consistent data. And then this is really kind of orthogonal to the I/O acceleration part, but it's kind of cool. It's just a Dragon system from Attica Genome. It's a FPGA-based computational accelerator. It's targeted specifically at life sciences and bioinformatics computing. They've been getting a lot of traction with different research institutes, and so we wanted to make one of these available to our researchers, mainly for like quick turnaround genome studies, genome analysis studies. So like you might find in a clinical research or precision medicine types of applications. And so we have a lot of really interesting life sciences research that goes on the UC San Diego campus. But these were the 4 science use cases that we highlighted in our proposal. So we've got the Gleason lab, which uses genome analysis to investigate causes and treatments for pediatric brain disease. We've got the Yo lab, which does RNA-seq analysis to understand the role of gene expression and development and aging. We've got the Human Vaccines Project, which we're collaborating with the Craig Venter Institute on. That part of what they're doing is trying to characterize what they call the human immunome. And that's it requires some data-intensive computing and a lot of I/O. And then the CBAT lab, which does, again, genome analysis to study neuropsychiatric disorders like autism. And so, that's all the good stuff. The bad stuff is we actually just have got the IME system up and running in the last 2 to 3 weeks, and so we don't have any results for you yet, to report. Now, we did do earlier. We were able to take the IME software and deploy that on one of our Gordon flash-based I/O nodes and do some quick experiments with GATK, which is one of the major genome analysis pipelines, and we found some very good acceleration with what's called the haplotype caller or variant caller step with a very unoptimized, kind of IME deployment. So that gave us a lot of confidence that we're going to get some good results. So, this is going to be something of an experiment, but it's an informed experiment, and we know that this is going to be a great technology, and we're going to, get some really productive scientific computing out of it once we have it fully up and running. And that concludes my presentation.
Thank you, Ron. Any questions for Ron? Yeah, we got a question in the back here.
You mentioned something along the lines of that clients can, cache working sets on the IME flash layer. Is that something that you expect the IME software to sort of handle automatically in terms of most recently used, or is that something you expect your users to have to tell the system they want it to do?
That's what we sure hope the IME system is going to do for us. So, I don't know if any of the DDN folks may want to chime in on that, but
Mr. Coomer.
Yeah, so if you write out into the IME cache layer, it'll stay there. If you want to deliberately push it back to the parallel file system, you can give it an API call and it'll do that. If you fill up the cache, then it'll hit a threshold and it'll automatically start pushing down excess clean data down to the backing file system. So basically, the answer is yes, it'll cache it automatically.
Thank you.
All right, one other question here in the front.
So what was the reason for choosing Lustre instead of GPFS? And one second question, what's the cluster software you're using for management software?
The reason for, we've got a big multi-year investment in learning about Lustre. So, that's basically what we've been deploying on all our HPC systems for the last, well over 5 years now. So it's climb the learning curve there, and no immediate plans to get away from that. And we're using the Intel distribution.